the spring weather starts to warm up in Seattle, the Picardo community farm, known as the Pea Patch to Wedgwood residents, bustles with activities. While local gardeners tend to their small farms, the warmer weather also attracts students from Edmonds College to the Pea Patch. Instead of bringing gardening equipment, the students come armed with butterfly nets. The students are in search of cabbage white butterflies, scientifically known as Pieris rapae. The insect is found worldwide and is considered a pest that feeds on leafy greens, which gardens and farms like the Pea Patch have an abundance of. The conditions need to be right for ideal butterfly hunting. The wind must be minimal. The temperatures cannot be too hot. No matter the conditions, the Edmonds College students are bound to have a good time chasing the prizes that await them at the farm. The students are earning extra credit by gathering cabbage white butterfly samples to bring back to the lab located on campus. At the lab, students and staff will study the butterflies from egg, larva, pupa, and adult stages. The goal is to see how temperature changes affect growth rates.
So the way we started working on cabbage white butterflies is that about 25 years ago, I was an undergraduate myself. I had just recently graduated, and I was looking for a job, and I ended up working as a research tech in the Kingsolver lab at University of Washington. And my job was to run experiments on cabbage white butterflies. We were at that time looking for the effects of temperature on growth rates of Pieris rapae, the cabbage white butterfly. I returned to working on cabbage white butterflies because I wanted the students to focus on one model system. And so I started to reach out to my colleagues who worked on cabbage white butterflies, and they had been writing a grant and asked me to join them because they thought that adding the undergraduate research component was a great aspect of a broader impact of the research that they were doing. And also, in addition to that, because I was the one who had done the research on the project 25 years ago, I created the opportunity to actually have the same person running the research that ran the research 25 years ago. So I knew how it was run down to the details of protocols, et cetera. So it allowed us to really have that consistency of repeatability between the project 25 years ago and the project today.
The WARP project, it's called the Washington Resurvey of Periodids because it is a resurvey of the data and the research that Dr. Schlichter did 25 years ago. And so far, it is a bit early, but there is some evidence to prove that based on the comparison between the historical data and what we have now, that the cabbage white butterflies are beginning to apply adaptive strategies to evolve and essentially kind of beat the effects of climate change.
Working for the Pieris rapae has definitely been interesting. I had no idea about this organism. I've always seen outside this little white butterfly flying around, but I didn't really have an attachment to this, right? And so to see that it was this species and working with it, there's definitely now a better understanding when I'm looking in my backyard that I know about this and how it can be affected. So a lot of the research that they have going on in the lab is understanding how global climate change is going to affect a lot of these types of organisms, because it's not just Pieris rapae, it's all pollinators, all ectotherms. And so I think understanding this organism really gives you something to look at into the future on all the different elements that go in and what can affect this organism as a whole.
Experience is really valuable because we're working in a lab setting and we're working on all these procedures, and a lot of it is really independent. We had to come up with our own research proposal and our own methods. And there is guidance from Dr. Glenn, but for the most part, a lot of the reins are handed off to us. So it's really valuable lab.
In June, students who conducted undergraduate research in STEM fields like chemistry, physics, mathematics, and engineering joined the biology students for a research symposium at Hazel Miller Hall. There, they eagerly share their findings with their peers, visiting scientists and curious community members.
So we wanted to see if, as temperature increased, they would consume more, which would be more detrimental to agricultural plants. We based ours over the past research, and we saw that past research usually they peaked around 30 degrees, and recently in our experiment we saw they peaked at 35 and 38. So that means they're like conforming more to current day's climate than past.
Course-based undergraduate research experiences, or CURES, here at Edmonds College mean that all students have access to a research experience and not a select few who have the opportunity to go and have an internship in another location. Mary Whitfield and myself received a NSF ITYC grant, which is Innovation in Two-Year College Grant, for $500,000 to support undergraduate research here at Edmonds College, and more importantly, to support external validation of undergraduate research, which is essentially things like the Undergraduate Research Symposium, as well as other opportunities like service learning and collaborations with universities. And so we hope that this will increase the presence of undergraduate research at Edmonds College and increase the opportunities for our students to pursue careers in research.
The Edmonds Biology Department was like really crucial to my success, I would say. Everyone fostered a very supportive environment. They gave a lot of mentorship. They were all very approachable. And the content that I learned from the biology department was just excellent. It really helps like now that I'm at UW to like reflect upon the skills that I was taught at Edmonds. Cabbage White Butterfly Lab here in Edmonds like led me to rediscover and really like fuel and solidify my passion for biology, and that it gave me the skills that I needed to have to be a resilient scientist and a resilient like biologist in general. And also like when you go to University of Washington, you have to do like an application for your major, which I did the molecular cellular developmental bio, and I also talked about research there. And the fact that I was introduced by Dr. Gwen to Dr. Buckley and was able to participate in like field research really helped, I think, give me an edge to get into the biology major itself.